Hey, this is Jonathan from Chegg Tutors. This lesson is going to be about interference in genetics that occurs during crossover events and meiosis. This is when a crossover event in one region will affect the probability of a crossover event taking place in a nearby region. And you can think of this as when a chiasma is formed when the genetic material is exchanged between two chromosomes. It kind of affects the ability of another chiasma to form because it kind of gets in the way. So you can't form another one nearby because you're already forming one in one region. So let's take a look at one example. Let's say we have genes A, B, and C. We have two homologous chromosomes here, and we have two alleles for the same genes. So if we had a crossover event that occurred between genes B and C here, and some interference took place, this would affect the probability for a double crossover event to take place, where another crossover takes place here between A and B. So a crossover here affects the ability of a crossover to take place here. So it just interferes with that probability of two crossovers taking place. So interference can be calculated using something called the coefficient of coincidence, which is defined as the number of double recombinants found in the progeny. This will be our experimental results, divided by the number of double recombinants expected in our progeny. This is if no interference took place. So if the coefficient is 1, where they're both the same, there's no interference. And you can think of it as just how well did our experimental results coincide with our expected results. So interference is then defined as 1 minus the coefficient of coincidence. So if we had 0, that means interference is 1 because we had no double recombinants found, which means that the crossover in one region really affected the crossover in another region. So let's look at an example. So again, we have genes A, B, and C, and we're going to first cross a homozygous dominant for all three genes with a homozygous recessive for all three genes. So our F1 will all be heterozygous, and the F1 chromosomes will look like this, with big A, big B, big C on one chromosome, little a, little b, little c on the other. Okay, so then we're going to test cross the F1 progeny with a homozygous recessive for all three genes, and we will get our results. Okay, so this is our results formed from the cross of a heterozygous F1 with a homozygous recessive in our test cross. And remember, we can kind of neglect the chromosomes inherited in the F2 from this individual because the only gametes it can form are a, little b, little c. That's why you don't see it in the genotypes here, because they all will have this little a, little b, little c. So these are the most abundant, so these are our parentals. These are going to be our double recombinants because they're the least abundant, because double crossover events happen less frequently than single crossover events. So DCO for double crossover. And these are all our single crossover events. So first we need to get our gene map. Let's say here's our chromosome. And to do this, we have to first find what gene is in the middle. So we do this by comparing the parental genotypes to the double crossovers, because whatever gene switched in the double crossovers, that will be the gene in the middle because it's the same thing as the two genes on the outside switching. So in a double crossover event, the two genes on the outside will switch, which is the same result as the middle one switching. So if we compare big A, big B, big C to this one, we see that the A's switched here when you compare it to the parentals here. So A must be in the middle, so we can just put B and C here. Okay, so now let's find the distance between these genes. So let's first do the distance between B and A. So first we look at the progeny and see where B switched. So here, in these two progeny groups, the B switched. So this is where a crossover took place between B and A. And remember, in the double recombinants, we also had one crossover that took place between B and A. So we need to count those two. So the total number of crossover events that took place between B and A is 90 plus 80 plus 10 plus 5, and we divide this by the total number of progeny, which is 1,000. Multiply it by 100 to get the percent recombination, and you get 18.5%, which is the same thing as 18.5 centimorgans or map units. So that is the relative distance between B and A. So now we have to do the same thing for A and C. So here, a crossover event took place between A and C because you can see that the C switched. And we also had one crossover event taking place in the double recombinants. So let's do it the same thing for A and C. We have to do the total number of crossover events that took place between A and C divided by the total number of progeny. So that's 55 plus 60 plus 10 plus 5 divided by 1,000, which is, and then we multiply by 100, which is 13% or 13 map units. So now we have our genetic map. Okay, so based on our genetic map, what is the expected number of double recombinants in the progeny? Well, a double recombinant means that a crossover event took place between B and A and C. So what's the probability that a crossover will take place between B and A? It's 18.5% or 0.185. What's the probability of a crossover event taking place between A and C? 13% or 0.13. The probability that both events will happen is the product of these two probabilities, which is equal to 0.024. So if we have 1,000 progeny, we can get the expected number of double recombinants by multiplying the probability that a double recombination will take place by the total number of progeny. So 0.024 times 1,000 gives us 24 expected double recombinants. But we only had 15, so clearly there's some interference going on. So let's first calculate the coefficient of coincidence, which is just equal to the results found in our experiment divided by the expected results. So it's 15 divided by 24, which gives you 0.625. And now to get the interference, we do 1 minus the coefficient of coincidence, so 0.0625, and this gives us an interference of 0.375. So that's how you calculate the interference between two crossover events taking place. I hope this helped, and my name is Jonathan, and I'm on Chegg Tutors.